welcome back to Well, That's Interesting, I knew it was going to be about my innards edition. Today is episode 219. Let's talk about the oldest naturally preserved human brains discovered so far. My friends, last week I had the honor of drinking too much and sharing a delightful TED-Ed talk whose experiment showed how our brains relay signals. And I hinted that this week's episode may be related. And boy howdy, it sure is. But I threw in a curveball. In the first half of the show, if you're like me, on occasion you find yourself Googling what's the oldest piece of preserved human brain material, and you get yourself an answer. But if you're like me, you dig a little deeper, and you find yourself a few more answers. The first answer you get is something called the Heslington brain. And even though the holiday season itself is over and preserved in memory, this brain is a gift that just keeps on giving. Way the fuck back in 2008, in Northeast England, it is believed the best preserved ancient brain in the world was discovered. A single lone skull was discovered in a waterlogged pit during an archaeological dig commissioned by the University of York. Now, finding a head may be disturbing for some, but for the archaeologists combing the 20 acres within Heslington, Yorkshire, well, they were expecting it. This place is a well-known place filled with past rituals. Rituals from over 2 millennia ago. So finding nothing but a human head was just another day. The team was excited for the find, but they had no idea just how special it was until the skull was transported within a block of soil to the Finds Laboratory of the York Archaeological Trust. My friends, it is super early in the episode to send you on over to our social media stuffs, but whatever you have in your hands right now, be it the wheel of a car or a defibrillator, just drop it. Pick up your phone and tap on today's post. The first image you're going to see is a purple latex glove, and in the hand of that purple latex glove is a piece of a human brain. And I am so sorry to get graphic, but my friends, that is shiny and plump. My lord. It looks as though it was plucked right out of my noggin, but thankfully it wasn't. When Finds Officer Rachel Cubitt was performing her kick-ass duty of cleaning that skull, one chunk of mud wasn't a chunk of mud at all. It was this hefty piece of human brain, with what the researchers described had the consistency of tofu. Yeah, my young business goose, if you're a long-time listener of this show, you know that soft tissue, especially our gooey brain parts, they don't survive very long in the wild. They're the first to decay or be scavenged. So, to not only find this sizable portion, but in this condition, it's nothing short of winning the archaeology lottery. The question of how in the holy hell brain matter, 2,600 years old, survived in such a state, it was a mystery until recently. We will get into the theories of how this piece survived. We will get into the cutting-edge technology used to finally be able to look at this brain on a molecular level, and we're going to find out who this person was and how they died. And spoiler, it was fucking terribly. My friends, this is a tale that goes back to 600 BC, and you'll be on the edge of your seat every moment. Then after the break, University of Oxford forensic anthropologist Alexandra Morton Hayward released a study in March of 2024 that told researchers around the world to hold her formaldehyde. She and her team rounded up an archive of more than 4,400 human brains preserved in the archaeological record, and guess what? They found evidence of naturally preserved human brains up to 12,000 years old. 12,000 years. Here's one fun example. In 1982, construction workers uncovered dozens of 8,000-year-old human skeletons in a pond. Where else but in Florida? Now, what is known today as the Windover Archaeological Site, archaeologists were shocked to discover intact brain tissue inside 91 of the skulls. Now, the tissue wasn't as pristine as the Heslington brain, but some were intact enough to identify contours and extract DNA. We are going to scrape into Alexandra's study and learn about some other ancient brains. We're going to learn about the different types of environments that preserve them. And we're going to talk about what her team calls, quote, the fifth mechanism. Not the fifth element, but mechanism, which is a mysterious combination of things not yet known to science that helps brain matter survive. And spoiler, it's what helped the Heslington brain survive, and we're still trying to crack the code. In the meantime, hi, I'm Jill Chacha. And if this is your first time listening, welcome to the flock, my bouncy business goose. To begin, I'm going to hose down the old time machine and pluck off the New Year confetti because we're all squeezing in and heading on over to the forgotten age of 2008. Our party wagon has just landed 3 kilometers, or just short of 2 miles, southeast of York's city center. And if York sounds oh so familiar to you, my deja-vuing business goose, you are absolutely right. Back in the wonderfully disturbing Halloween episode of episode 212, let's talk about maggots hatching from people's foreheads and the deepest cave in Australia so far, we took a trip to York, where in 2019 a woman found herself at the York Teaching Hospital asking a tiny but mighty team of infectious disease specialists to look at a bump on her head. If you haven't had a listen, I really think you should. As you could tell by the title, a lot happens. And she's not alone in the maggot in the head department, so my God, what a day that was. And what a day we're about to have here, once more, this time in 2008. As for my fellow geographically challenged Americans who need a little refresher about York, don't worry, I've got you. Everyone, please imagine England. Perfect. Now point to the lower right-hand corner. Okay, there is London. Now move that finger up quite a bit. Keep going. Up, up, up. Perfect. Right there. And then move it to the left. Excellent. There is Manchester. Now one more time for the last time. Just move it up just a smidge. And to the right. Here we are, in a walled city in northeast England that was founded by the ancient Romans, a city called York. Now, in ye old episode 212, I mentioned York is home to a 13th century Gothic cathedral, aptly called York Minster. It's adorned with medieval stained glass and 2 functioning bell towers, but I gotta be honest, a building from the 1200s is relatively young for this city. When the Romans rolled in, it was 71 AD. Yes, a year with only double digits. And for some unfortunate reason, they called the area Aboricum. You heard me, Aboricum. Do you want to say it? I know you want to say it. Okay, let's say it together. Aboricum. Fantastic. Now, this may come as a shock, but Aboricum is also relatively young for this area. Archaeological evidence suggests that Mesolithic people settled in the region of York between 8,000 and 7,000 BC. That means humans have lived, laughed, and loved here for 10,000 years. During this time, agriculture developed and certain animals became domesticated. Cereal grains like barley and wheat were cultivated, and many of the Ice Age megafauna, like the things that hunted us, including cave bear, cave lion, and the last of the saber-toothed cats, they went extinct. This entire combination led humans to finally settle, a first in our young species history. This also meant, for the first time, we left a lot of shit behind. Now, in what is present-day Heslington, about 2 miles southeast of York, where we are, under the summer sun in 2008, you could imagine the sweaty excitement of researchers of the York Archaeological Trust, who were digging away within a vast 20-acre excavation site. Now, get this, my spooky business goose. Since 2003, the dig was commissioned by the University of York, who, on this ancient, clearly haunted land, chose to build its new campus here, on this. And they would call it the Heslington East Campus. So, if you or someone you know, for the love of God, if you go and you attend Heslington East Campus, please tell me, for the love of God, if you experience anything weird. I want to know about it. Anyway, this area, as I've said, was already found to have been a site of well-developed permanent habitations. So breaking ground and building something new on what could be a burial site or a monument, it would be catastrophic. These 20 acres had to be swept clean. So for the university and the trust, this was a win-win situation. But neither of them knew just how much the trust was going to win out on this one. The remains of prehistoric fields, buildings, and trackways were discovered, dating from the Bronze Age to the middle of the Iron Age, aka from 2500 BC up to 100 BC. According to the area's wiki, excavators found a number of circular features, which they interpreted as the remains of roundhouses. And not too far off from these homes were pits. Let's get into them. Around a dozen or so were found, and wouldn't you know it, when researchers gave a look-see into them, they were like, yeah, those are ritual pits. For sure. For you see, quote, some pits had been pierced with a single stake, while a number of pits included burned cobbles of a local type of stone. End quote. In one pit was the headless body of a deer. In another, a red deer antler was found. However, in one waterlogged, muddy hole, forgotten until late summer, August of 2008, a single human head was unearthed. Archaeologist Jim Williams had the honor of seeing the back of a human cranium. The head was found face down in a ditch, with no evidence of another part of a human body nearby. Jim did what any one of us would have done in this situation and asked someone else to pick it up. He called upon his colleague and friend Rupert Lotherington, who in best friend fashion picked it up and went the extra mile of packing it away in a block of the mud it was found in, and he sent it off to the Finds Laboratory of the York Archaeological Trust. Now, my friends, I don't know what your day job is, but for Finds Officer Rachel Cubitt, it's cleaning bones. And today, it was Rachel's time to shine. The skull was accompanied by a jawbone, and the first two vertebrae were still attached. However, no sign of hair, skin, or any other external soft tissue were to be seen. Please put a disturbing pin in all of this. We will get back to it. When it was time to look inside the skull, she noticed something loose. When she peered through its base, she saw that it contained a yellowish substance, and it was right then, she had herself a eureka moment. From an interview with Nels in December of 2008, Rachel said, quote, "It jogged my memory of a university lecture on the rare survival of ancient brain tissue. We gave the skull special conservation treatment as a result and sought expert medical opinion." End quote. My living business goose, the skull, and its unusual contents were put into cold storage and examined using a variety of medical and autopsy techniques, and I bet you want to know what they discovered. Of course you do, that's why you're here. Drum roll, please. Thank you in the back. At some point, the skull belonged to a man aged somewhere between 26 and 45 years old at the time of death, which most likely took place in his mid-30s. As to what year and how he died, you might want to take a seat. radiocarbon dating found that the man passed sometime between the 7th century BCE and 5th century BCE, aka 673 to 482 BCE. So this was an Iron Age man. But he might not have been British. Researchers whipped up a mitochondrial DNA test that revealed he belonged to haplogroup J1D, which, according to a study on this brain published in the Journal of Archaeological Science, had not previously been seen in Britain, in either living or dead individuals. In fact, J1D has been identified in only a few people from Tuscany and the Middle East. Perhaps his not being a local explains the following. He died from traumatic okay, bear with me. He died from traumatic spondylolisthesis, which is a fancy-ass difficult way of saying a complete fracture of the spine, usually associated with death by hanging. But don't worry, it gets worse. Time to unpin that vertebra pin for me, because according to the Heslington Brain Wiki, quote, shortly after death, his head and upper vertebra had been severed from his body, almost surgically, using a thin-bladed knife. Ugh. Although there is evidence from other sites of trophy heads in the Iron Age societies, it appears that the head was immediately deposited in the pit and buried in fine-grained wet sediment. Also, side note, please put a hefty pin in that too. Continuing the quote, the reason for the killing is unclear, but archaeological context suggests that the man may have been killed for ritual or sacrificial purposes. End quote. So, in sum, he was killed ferociously and torn apart just as quickly. As to where the rest of him is, it's anyone's guess. But we have his head, and boy howdy, what's inside is more than anyone could have expected. His brain had shrunk to about 20% of its original size, and chunks of it survived the decaying process. I repeat, it avoided the decaying process for 2,600 years. If you haven't yet, drop whatever it is you're holding that isn't your phone, it's not important. Head on over to our social media stuffs and tap on today's post. There you'll see a series of photos of this man's brain, or what's left of it. Again, not to get even more graphic, but y'all, this looks kinda new, fresh, recent, etc. So, if coincidentally a part of you is wondering how in the holy hell is that possible, and you're thinking it's gotta be more than just the environment, since only the brain tissue was found. If you thought any of this, you are not alone. So join me, will you, once again in December of 2008, when news of the Heslington brain hit the media. Rachel and her colleagues were landing interviews left and right, and it just so happens, at the same time, the awesomely named Dr. Axel Petzold, a neurologist from University College London's Queen Square Institute of Neurology, Dr. Axel needed a distraction. I fucking kid you not, and simply put, it was the morning after his lab's Christmas party, and Dr. Axel was hungover as fuck. Telling Rachel Funnel of IFL Science, quote, I just couldn't face life, end quote. We've been there, dear Axel, and just like Axel in desperate times, you take desperate measures. He went on to say, quote, I normally wouldn't start the day listening to the radio, but I was just sitting there with a coffee, slowly getting started, and I heard someone talking about the discovery. And I started thinking about it. End quote. My friends, the miracle of alcohol and the burden of hangovers is what we can give thanks for, because it was these two things that joined together Axel and the team at the Fines Laboratory of the York Archaeological Trust. For you see, Axel spent his career pioneering research in two types of filaments in the brain, neurofilaments and glial fibrillary acidic protein. I eventually nailed that. Anyway, they're called GFAP for short, which we're going to call them that. Anyway, long story short, these two act like scaffolding to hold the brain together. And he suspected that these proteins may explain why and how this brain survived. By 6 p.m. that same day, Axel connected with Dr. Sonia O'Connor, the researcher in charge of investigations into the Heslington brain, making this one of the most productive hangovers in human history. You will see why in a minute. After scoring a sample of our Heslington brain, Axel spent the next decade, 10 years, performing analyses and experiments trying to figure out how this brain doesn't look a day over 600 BC. Needless to say, technology grew exponentially every year since 2008, and with it at hand, finally, we now have an idea of what happened in this noggin. First, let's talk about your noggin, and mine, and everyone else's. Under average circumstances, if our bodies are left to decay, fatty, spongy organs such as the brain, which is about 75% water, these are the first to go. And one way they go is boy. Did something happen to my brain? And one way that they go is autolysis. It's a fancy-ass word that sums up the process by which internal enzymes destroy and break down cells. Something here with the Heslington brain, well, it put a stop to that. For some reason, those enzymes were prevented from tearing down that scaffolding we talked about just a moment ago. They could not unravel the organ's infrastructure. Thanks to cutting-edge technologies, such as mass spectrometry, in 2023, Axel and his team did what any one of us would do if given the chance. They watched tissues from both the Heslington brain and a modern brain decompose over an entire year on a molecular level. And what they saw left them speechless. The Heslington brain remained intact. So much so, 800 proteins could be easily recognizable, and for fuck's sake, some were in such good enough condition that they were able to elicit an immune response when prompted. Remember those neurofilaments, those GFAPs? Well, they're typically found in greater concentrations in inner areas of the brain, but in the preserved Heslington brain, researchers found the opposite. More filaments were in the outer areas of the brain. Somehow those proteins survived, and they did the complete opposite of breakdown. They folded in on themselves, forming, quote, tightly packed aggregates, which are more stable than those found in live brains, end quote, from the aptly named study, Protein Aggregate Formation Permits Millennium-Old Preservation. Y'all, I'm going to need some of those aggregates. From the aptly named study, Protein Aggregate Formation Permits Millennium-Old Brain Preservation. In fucking sum, my gently wrapped business goose, this dude's brain created its own shell casing to prevent decay. Now that's damn impressive. But I can see your reaction from here. Your face says, yeah, that is impressive. But we said things like brains decay from the inside out. What slowed that down? How do we know? And why? Why did these filaments seize up to begin with? That's what I want to know. Answer me. Amazing questions, and that's perfect timing. Well, thanks to these aggregates and the wet, anoxic environment in which the skull was found, that could have prevented microorganisms from getting all up inside the skull, and that takes care of the outside part. As to help explain some of the inside part, I have great news. We can only guess. Quote, the human body tends to decay from the inside out, consumed by a post-mortem swarm of bacteria from the gut, which spread around the body via blood from the alimentary tract. In this particular case, the head was severed from the alimentary tract and drained of blood, so the intestinal bacteria did not have an opportunity to contaminate it. The precise mechanism by which the Heslington brain was preserved is unclear. End quote, from the Brains Wiki. My friends, it's believed, along with the unique environment, the unique situation, i.e. the swift death and decapitation, played a massive role here. But there is an unknown element, and it's vital. It could be, quote, that perhaps an acidic fluid seeped into the brain, end quote, as noted by ucl.ac.uk, or, quote, even a hidden health condition was triggered, one that produced dense clumps of proteins in neural tissue, end quote, from Rodrigo Perez Ortega of Science magazine. If you will, put a gigantic pin in this unknown element, as we will get back to it after the break. In the meantime, other than a very impressive brain, you might be wondering, what is the big fucking deal? Why do we need to care about something like this? Well, Dr. Axel has an idea. These particular brain proteins, their folding, or lack thereof, could have major implications on biomedical research. When the team had extra time on their hands, for real, they conducted an experiment to see how long it would take for a brain and its brain protein aggregates to unfold themselves. And they found it took a full year. This could imply that treatments for neurodegenerative diseases, that they may need to consider a more long-term approach to tackling harmful or, when creating helpful, protein aggregates. This includes treatments for diseases like Alzheimer's, Huntington's, and Parkinson's. Who knew that one ritualistic murder from 2,600 years ago might help someone someday soon? After the break, more great news. I have 5 additional brains for you, and one very excited human. We will meet forensic anthropologist Alexandra Morton Hayward and dive into her study, a study that breaks down the 5 ways a human brain can survive over thousands of years, like way more than 2,600 years. She and her team have several refrigerators worth of samples to prove it, and we're going to take a look at a few. That's the plan. Stay tuned.
This episode is brought to you by Indeed. We're driven by the search for better, but when it comes to hiring, the best way to search for a candidate isn't to search at all. Don't search, match with Indeed. Use Indeed for scheduling, screening, and messaging so you can connect with candidates faster. Listeners of this show will get a $75 sponsored job credit to get your jobs more visibility at Indeed.com/SBO. Terms and conditions apply.
And we're back. We are so back. And my friends, I'm going to ask you to slap on your finest latex gloves, because we're about to handle some brains that have seen some shit. Now, I can tell by your expression you are also excited about this, but no one is as excited as Alexandra Morton Hayward, who is now a forensic anthropologist over at the University of Oxford. If you drop your sandwich or child and pick up your phone to tap on today's post over at our social media stuffs, swiping past the photos of the Heslington brain, you'll see Alexandra absolutely fucking beaming and holding up a naturally preserved 1,000-year-old brain of an individual excavated from a churchyard in Belgium. This is just another day for Alexandra, and it was her day-to-day that inspired a recent paper. One titled, "Human Brains Preserve in Diverse Environments for at least 12,000 Years," as published in the March 2024 issue of the Proceedings of the Royal Society B. You know the one. But before we dig into that, let's meet Alexandra, who started her career with people's heads first as an undertaker, telling Andrew Curry of Science.org, quote, "I loved it. One of the things that intrigued me most was decomposition. Just as we're all different in life, we all decompose differently in death. A lot seems to depend on how you lived and what you died from. End quote. Well, my exhausted business goose, Alexandra's fire in the belly for decomp fueled her PhD, which led her into a smothering avalanche of literature citing hundreds upon hundreds of cases where a human brain had somehow remained intact after burial. And we're not talking a few days here. I'm talking a few millennia. It became her calling to round up this information, to find these samples, and create a catalog like no other. Enter the paper I just mentioned. Let's get into it. My friends, Alexandra and her colleagues collected an archive of more than 4,400 human brains preserved in the archaeological record, all sourced from every continent except Antarctica. And with this information, they, quote, established the prevalence, persistence, and diversity of preservation types. Let's do that again. Established the prevalence, persistence, and diversity of preservation types. End quote from the study. I should have put that on one line. It was on 2 pages. Anyway, very long, pokey, and proby story short. The team identified 5 types of brain preservation: dehydration, freezing, saponification, tanning, and soon to be your favorite, a 5th mechanism, simply classified as unknown. Let's meet some extraordinary examples of each, shall we? Now, if you're not too busy examining someone else's head, take a look at today's post over at our social media stuffs and swipe along with me. I've got one hell of an impressive slideshow for you, as provided by the team study. I am so excited about this. Okay, let's start with the most recent brain, and the one whose preservation type probably made you say, what the fuck is that? Saponification. Don't worry, I've got you. My friends, back in 1936, a little thing called the Spanish Civil War occurred, leading to the usual atrocities we see, something that we humans are so stellar at performing. At least 45 bodies were eventually excavated from a mass grave. And even though some skulls were, quote, highly fragmented by gunshot trauma at the time of death, end quote, their brain matter and a significant amount of their body's soft tissue avoided complete decay. Why? Well, a theme you've heard before. A quick death and a supportive anoxic environment that in this case turned the brain into a, quote, grayish-white mass of variable consistency, ranging from hard and crumbly to a soft, paste-like texture formed by a hydrolysis of triglycerides, end quote. Ooh, that's a fancy way of saying that the fatty acids in their bodies reacted with nature stuff like disodium and magnesium ion to produce soap. Yes, a soap. Like soap, my friends, a substance that goes by the name grave wax. It encased their tissues and led to preservation. This is called saponification. So the next time you take a shower with a bar of soap, please think of me and this episode. Anyway, moving on to sample number 2, and it's a doozy. May I present the brain of an Incan human sacrifice? Delete. May I present the brain of an Incan human sacrifice found on top of a volcano? Yeah. Y'all, I don't know what's going on with me tonight, but... Moving on to sample number 2. It's a human sacrifice that was found on top of a volcano in South America, and it dates back to 1450. This is actually one of 3 individuals that were found on what is still the world's highest archaeological site, Yuyayaco. Oh man, I nailed it. How come I can say that, but I can't say everything else? Yuyayaco is a dormant volcano on the border between Argentina and Chile. And if this sounds familiar, boy howdy, you know it. We covered these child sacrifices before. Of course we did. Check out episode 089 or episode 197 if you haven't. Those were good times as well. Today, though, This is a CT scan of one of the three individuals, and it leaves me absolutely speechless. An extraordinary amount of brain material has survived here thanks to the arid climate and a countless series of freeze-thaw cycles. It is nearly whole. It is absolutely insane. That is all I could say. And next under the category of tanned, an archaeological pop star, this is what's nicknamed the Tolland Man. The Tolland Man. Mand? Not M-A-N-D, girl. Tolland Man. This individual was unfortunately hanged and dumped in a bog 2,400 years ago, aka 240 BC, in what is now Denmark. And we know of his fate because he was discovered 8 feet down in a peat bog with a rope around his neck. And thanks to that environment, his nearly intact body revealed even more. The acid of the surrounding sphagnum moss put his soft tissue through a tanning process similar to that used for leather. So he became a hide of sorts. And as you could tell from the radiograph here, performed in 1950, this process also saved his brain from microbes and invading bacteria. Now, 240 BC is old, but we can do better. My glowing business goose. Swipe past this, and you'll behold one of about 500 brains. One of 500 brains from 6,000 to 3,000 BC. I'll say that again. This is up to 6,000 years ago, uncovered from Upper Egypt within the shallow sandy graves along the Nile. These were simply dehydrated with, quote, non-reducing sugars like disaccharides, replacing water around hydrophilic amino acids, creating a glassy state, end quote. Naturally. So they were just dehydrated. Now, it's impressive, kind of, but we haven't even started. Please, swipe past this, and you will see the piece de résistance, a sliver of brain that is truly remarkable for a number of reasons. And it just so happens to be our last preservation type. As described by Alexandra herself, quote, "What I'm really interested in is brains that are preserved through this unknown fifth mechanism. Although we assumed watery conditions would speed up decay, these brains are usually excavated in wet environments. But not always. They're found in burial mounds and sunken shipwrecks, at the bottom of wells, and in lead coffins. They're retaining their shape and microscopic architecture when the rest of the body's soft tissue has completely disappeared, which is amazing. I have over 570 brains of this unknown type in my lab right now. 4.5 fridges full of brains from different European countries. And I have no doubt people will let me know about more when this paper is published." End quote. From the interview with science.org. Yeah, my friends, not only does Alexandra have 4.5 fridges full of brains like these, but just as with the Heslington brain. And unlike the other samples we just talked about, no other soft tissue was found on the body. Only the brain survived. In the example provided by the study, here we have, once again, a severed skull, but one which had been mounted on a wooden spike found beside a lake in what is now Sweden. What a visual. 8,000 years ago, in 6350 to 5000 BC, this person was, let's say, made an example of. And to this day, as you can see in the circled area in the photo here, quote, "A resilient hydrophobic mass is largely the only soft tissue amongst skeletal remains," end quote. My friends, 1/3 of reported brains in this study, that's over 1,300 out of 4,400, exhibited this fifth mode of preservation. This begs the question, fucking how? Alexandra has a fantastic theory, as she told Science.org and NewScientist.com, quote, "My belief, based on the evidence, is that it's something particular to the central nervous system because of the brain's unique biochemistry. There are fats in the brain that aren't expressed anywhere else in the body, and sulfur-containing proteins that are a part of the brain's signaling mechanism. I think all that is fusing together to form a material that can preserve for a really long time. Maybe substances such as iron catalyze the formation of crosslinks between proteins and lipids, forming more stable molecules that resist degradation. Oh my God, folks, today is the day. Continuing the quote, "The nature of the proteins and lipids found in the brain, or their ratio, might be the key." End quote. So, is it a combination of the type of death, the surrounding environment, the fats, sulfur and iron in the body? If it's a combination of all of those, that sounds good to me. I am totally down with that. But to say for sure, more analysis needs to be done. And thanks to Alexandra and her team's study, we now know before you disregard a skull, you might want to triple check if there's some brain in there. If so, its DNA, its proteins and folds can show us so much, and it could save lives. Oh, I almost forgot to mention, you know how Alexandra learned that there's still copious amounts of iron left in these types of preserved brains? Well, one day, when she was handling a sample, she decided to put it through an MRI machine. She told Michael LePage of New Scientist, quote, "It was a terrible mistake." She didn't go into details, but I assume it exploded a little bit. Thank you for listening, rating, subscribing, getting through all of my inabilities to speak. Thank you for that. Thank you for being patient. Thank you for telling your friends about human brains. You know, it's a great way to start the new year in a conversation while they're eating. Just go through all the different ways brains are preserved, how long they could be preserved, how these people died. It's really something. And an explosive thanks to the folks over at Airwave Media, the podcast network to which WTI belongs. If you love this show, and you do, you love the other podcasts in this family. And please, stay interesting.